This presentation is on acquired immunodeficiency syndrome, commonly known as AIDS. This presentation shall address the following things: the meaning of the term AIDS, the cross-sectional structure of the causative agent human immunodeficiency virus 1, its general life cycle, its mode of transmission, the different preventive measures, the different classes of drugs for combating the disease, and the different challenges associated with the curing of the disease. AIDS. It stands for Acquired Immunodeficiency Syndrome. It is the disease which is caused by the infection of the human immunodeficiency virus 1, which is responsible for causing damage to the immune system over a period of time, giving way to various opportunistic infections to take place. Some of the common symptoms associated with AIDS are opportunistic fungal infection caused by Candida albicans, respiratory illness like pneumonia and tuberculosis, intestinal illness like chronic diarrhea. Out of the many cancers, Kaposi's sarcoma is very closely associated with AIDS. Encephalitis, which is the inflammation of the brain. Meningitis, which is the inflammation of the tissue covering the brain and the spinal cord. One of the significant symptoms associated with AIDS is the huge reduction in the CD4 count. And this decrease in CD4 count is the hallmark of AIDS. Let us have an overview of the structure of HIV-1. HIV is an enveloped virus. The envelope is derived from the host cell membrane. The envelope has two viral proteins, GP120 and GP41. GP41 is a transmembrane protein. GP120 binds with the CD4 receptor of the host cell. The core is composed of P17 matrix protein and P24 capsid protein. Inside the core are two identical single-stranded RNA viral genome and enzymes, reverse transcriptase, protease, and integrase. This slide gives the schematic representation of the life cycle of the virus. Let us try to understand the different events stepwise. The first event is the binding. There is an interaction between virus GP120 and CD4 receptor of the host cell. This interaction brings about a conformational change on GP120. In addition to CD4 receptor, there must also be the presence of co-receptor, which is CXCR4 in case of T cell and CCR5 in case of macrophage. The co-receptor brings the two membranes closer. As the two membranes come closer, the stalk of GP41 pierces through the host membrane and bring the two membranes so close together that they fuse. The next event is the release of viral components. With the fusion of the two membranes, the core with viral RNA and enzymes enter the T cell. The capsid and the matrix are degraded by cellular enzymes. As a result, the RNA and enzymes are exposed to the cytoplasm of the host cell. In the process, the envelope remains on the surface of the T cell. Next step is reverse transcription. With the entry of the viral genome, the reverse transcriptase enzyme synthesizes single-stranded DNA using viral RNA as the template. It then later degrades the viral RNA. It then constructs complementary DNA to form double-stranded viral DNA. This process is opposite to the principle of central dogma, which states that information flows from DNA to RNA to protein. Therefore, this virus is known as retrovirus. During the process of polymerization, reverse transcriptase enzyme makes a lot of errors. This is because it has no proofreading ability. Lots of errors means lots of mutation. This point makes it develop resistance to medication and also makes it hard to develop vaccines against it. Next step is provirus integration. The enzyme integrase grabs hold of the double-stranded viral DNA and carries it inside the nucleus of the host cell. Inside the nucleus, it integrates with the host cell DNA. This establishes lifelong infection with the virus. At this stage, the provirus may remain latent or go for transcription. Following transcription is the translation process, which produces envelope proteins, and viral proteins are produced in the form of single-strand polyprotein. The final step is viral assembly and budding. The envelope proteins GP120 and GP41 are inserted into host cell membrane. Polyprotein is cleaved by the protease enzyme. It is cleaved into the viral enzymes reverse transcriptase, integrase, and protease. The structural protein forms the core. Finally, the viral RNA and enzymes are incorporated, and this assembly buds off from the host cell as a new viron. This new viron can infect new T cells. There are very many ways through which this virus can be transmitted. It can be transmitted through infected body fluid. It can be transmitted through unprotected sex. It can be transmitted by sharing injection needles. During childbirth, it can be transmitted from mother to child through blood, amniotic fluid, and vaginal fluid. It can be transmitted from infected mother to child through breast milk. It can also be transmitted through contaminated blood, especially during blood transfusion. The different preventive measures of HIV transmission are use of condom, use of sterilized needles for injection, screening of blood for HIV before blood transfusion, and to prevent transmission from infected mother to child, antiretroviral treatment can be given to pregnant women. There are many classes of drugs to combat HIV infection. These classes are fusion inhibitors, which inhibits the fusion between the viral and the host cell membrane. CCR5 antagonist, which glues the CCR5 receptor and prevents it from attaching to the viral membrane. Nucleoside reverse transcriptase inhibitors, which prevents the elongation of the nucleotide. Non-nucleoside reverse transcriptase inhibitors, which glues the reverse transcriptase enzyme and prevents it from carrying out its work. Integrase inhibitors glues the enzyme and prevents its function of integrating the viral DNA with the host DNA. Protease inhibitors prevents the protease enzyme from cleaving the polyprotein. Use of single drug gives the virus opportunity to develop resistance against the drug. But it seems that when it's given in combination, it makes the virus harder to develop resistance. Therefore, drugs are given in combination known as HAART, that is highly active antiretroviral therapy. It should be noted that antiretroviral therapy does not cure but can only minimize the level of HIV so that the immune system can fight infection. A lot of challenges are associated with the treatment of HIV infection. The foremost challenge is the integration of viral DNA to the host cell DNA because it makes a permanent association with the host cell DNA. Moreover, there are a lot of side effects of the antiretroviral drugs used. Therefore, to come up with a suitable vaccine seems to be an answer to stop HIV infection. But again, there are a lot of problems associated with the development of vaccine. The difficulties are HIV infection remains latent for long before causing AIDS. Usually, vaccines are used to combat diseases and not infection. HIV mutates rapidly. Vaccines are usually the killed or live form of pathogens. In case of HIV, the kill form does not retain antigenicity, while the live form raises safety issues. Vaccine protects against infrequently encountered virus, while HIV may be encountered daily. Vaccines protect infection from mucosal surface of respiratory tract or gastrointestinal tract, while HIV infection is through genital tract. Usually vaccines are given to protect from re-encounter of the same pathogens. In case of AIDS, usually there is no recovery of the patients. To study the efficacy of any vaccines, animal models are used, but there is no suitable animal model for HIV. These are the references I followed to make this piece of presentation. I hope I have been comprehensive enough. Thank you.